But how do peptide bonds really form? If you take an organic chemistry class, you might be taught like, oh, it's an amino group and a carboxylic acid group, and they're joining together in this condensation reaction. You lose water. Oh, and by the way, that's not how it actually happens in your bodies. And so you're like, okay, well, how does it happen in our bodies? And so it turns out that it actually happens through several steps. And so first you actually activate an amino acid. You make it to make it more reactive. So you add it, react it with ATP. You add the AMP part on there. And then you're going to just, from then on, you're transferring that to other places. So you're going to transfer it to tRNA, which is going to give you the specificity you need to make sure you're adding the right letters, the right amino acids that correspond to the mRNA. And then you're going to, with the help of the ribosome, catalyze the transfer of a chain that's of peptide, previously formed peptide bonds, like so a peptide that's already forming. You're going to transfer that onto the incoming tRNA, which is charged with the amino acid. And so you do end up losing the equivalent of a water molecule, but you're not actually losing it as a water molecule, and you're actually losing parts of it along the way. And the reaction is pretty cool, and I wanted to kind of just walk people through it. So this is going to be another one of those really technical ones. And with like arrow pushing and stuff. And so I'm not going to explain things as much as I normally do. Sorry. But hopefully this post will help people like me, who always got confused when they're like, but this isn't how it really happened. Okay. So in chemistry, you might see something like this. So you have this 2 amino acids. And so they have that, this is the generic backbone, and then their unique side groups, which I'm just going to abbreviate R1 and R2. And I've tried to color code things so you can see where things go. So this is our first one, and so you can see we have this carboxylic acid group and this amino group, carboxylic acid, amino group. And what we're going to end up with is this, where we have them linked. This is our amide bond, which is, we call it a peptide bond in this case, but it's an amide bond. And we're going to call this reaction, let's see how this reaction could go and what we're losing. So if you look at this here, you could have imagined, so remember that the oxygen is electronegative, so it's pulling electron density away from this carbon. So this is going to be partly negative, and this is going to be partly positive. You have this lone pair of electrons on this nitrogen. This can act as a nucleophile. The electrons are going to flow up to the oxygen, get your single bond, and now you're have this weird tetrahedral intermediate. And this is going to be an unstable intermediate. You can see it's kind of crowded, and you have this separation of charges, and it's not very happy. So what you can get is you can actually have the oxygen, the lone pair is going to flow back to reform that double bond, and this OH is going to leave with a hydrogen as water. So this is a condensation reaction, or dehydration. You're losing water. You see, you have the two, you have this OH from your first and the hydrogen from your second, and all's good, right? Except things really aren't that simple. And so when you learn this reaction, they'll say like, well, this isn't actually how it actually happens in your body. And it's not. In our body, it's done on the ribosome, and it's done with the help of the ribosome acting as an enzyme, so it's going to speed up the reaction. And it actually happens first, though. It happens through several intermediates. So you're actually going to use ATP to charge your to activate the amino acid. So basically, we need to do like a substitution reaction. We're going to substitute out this OH group for this part. But to do that requires this part to leave. So it's a leaving group. And OH is a bad leaving group. And so when we do it in our bodies, we make this a better, we can activate this by making a better leaving group. So we're going to switch this out for AMP, and then that's going to be a much better leaving group. Because although, like, so if we have this, we can get this oxygen to react to things, and like add on to things, it's harder to get this group to leave. So, and we don't want to, if we get the oxygen to react to things, we're going to make a bond to the oxygen, and we end up with an ester. But we don't want an ester, we want an amide. We want to react directly to this carbon. And so to do that, we're going to need to activate this group and make this a better leaving group, so it can go bye-bye when you make the new bond here. So how we're going to do this is we're actually going to use ATP to get energy, and we'll still end up with that same final product. So we still will get a the loss of equivalent of a water molecule, but we're not going to lose it like this in this way. We're actually going to lose it kind of stepwise, and the oxygen is actually going to be lost during the tRNA charging step. So in addition to activating it with the, so basically our first step is going to be to activate the amino acid. So we're going to swap out this carboxylic acid group. We're going to attach it to an AMP from ATP, and then we're going to the rest of it's just going to be transferring it to other things. So we're going to transfer it to tRNA, then we're going to transfer it from that tRNA to another tRNA. That's with the growing chain. We're going to go, or that's the incoming tRNA. So you'll see, it'll make more sense in a minute, hopefully. But yeah, so I've tried to draw this out. So I'm not drawing out the adenosine every time because it's this big bulky thing, but this is ATP. And so imagine everywhere you see adenosine, it's this, this group. So you have this ribose sugar, and then this adenine, nucleotide base. And you have these 3 phosphate groups in a row, which are so this is very high energy. This is very unstable. These are all negatively charged. It's kind of hard to see, but these are all negatively charged. And when you have a bunch of negatively charged things next to each other, they're going to try to repel. So you have high energy in this. So if you can break these bonds, you're going to generate a lot of energy that you can use to do things, like to form the bond between the amino acids. Okay, so how are we going to swap out that this group for the AMP? So adenosine, this is adenosine triphosphate, and what we're going to do is we're going to attach adenosine monophosphate. So we're going to add just this part, but we're not even going to add that oxygen. We're just going to add that part. And how we're going to do it, we can do this because, so remember, these want to break. These bonds are going to be easy to break, and that's going to give us energy. These oxygen here, you have this negative charge, you have these lone pairs. It can act to attack this phosphate because, so even though this is really negative overall, the oxygens are pulling electrons away from the phosphate, from the phosphorus, and this is going to make the phosphorus partly positive. And so you have this negative, you have this positive, you have this lone pair, so you can get this attack. When you get this attack, then this is going to go here, and this, you have this leaving as this PPI, and you have your AMP. So we started with ATP, and we're only going to attach the AMP part, okay? But now we have this better leaving group. So this is a much better leaving group than just an OH would be. So this is a good leaving group. So this is going to be reactive. Okay, so now we have this step is called amino acid activation. We've activated this amino acid. We've given it the energy that it needs for the reaction to happen. And so for the rest of things, we're not actually going to add any more energy to get this reaction to happen. But we are going to transfer it, and we're not going to transfer it directly to the other amino acid. Instead, we're going to first transfer it to a tRNA. And so the tRNAs, this is going to provide our specificity. And it's going to kind of give it a handle to be used in the ribosome. So remember, you have your mRNA, and it's going to have like codons, so 3-letter sequences. So it's going to have like a ton of these, but they're read in 3-letter chunks called codons. And so you'll have one codon here, and then your tRNA is going to have the matching anti-codon on it. So the tRNA, like the name sounds, it really is an RNA. And so it's going to have this anti-codon, and then it's going to be attached to the incoming peptide. And so I guess this would be red in our case. So this is our incoming peptide, and it's on this tRNA. And so this is going to allow the specificity. So the amino acid activation part was activating the amino acid. The tRNA charging part, we're actually sticking the amino acid onto the tRNA. And this is going to provide the specificity we need to make sure that we're giving the sequence specificity. So the mRNA is specifying the sequence. The tRNA has the matching anticodon with the corresponding protein letter. So this is the codon is in the mRNA. The anticodon is part of the tRNA. And then this is the amino acid to be added. And all of these have to match. And this is how you get the specificity. So different codons will give you different amino acids to be added. And the tRNA is going to act as the intermediary between the two. So by charging the tRNA with the amino acid that we want to add, charging the specific tRNA that has the anticodon that matches the codon for that amino acid. So this is how we're going to get our specificity. And so how is it going to work? So like I mentioned, the tRNA really is RNA. So it's a form of transfer RNA. And so RNA, it has bases like this. They're not all adenosine, but so you have this ribose sugar and then one of these bases, which can be A, C, U, or G for RNA. And so it has, just like this we saw with ATP, you have these two hydroxyl groups. And so this is going to be the, so this is the 3' OH, and this is the 2' OH. And so this 2' OH is the one that ribose has, so RNA has, but deoxyribose, so DNA, does not have this OH here. And this is going to come into play later as to why the ribosome, which is RNA, is able to catalyze this reaction. And so the tRNA, so in that active site, so this is the 3' OH, so now it's kind of flipped upside down. But this is the 3' OH group, so this here. It's going to now react with this. So we have this oxygen with these lone pairs. You can get this nucleophilic attack onto this phosphate group. You can get this nucleophilic attack onto this carbon that you've activated. So here the oxygen is going to be pulling electron density, remember? So this is partly negative, this is partly positive, and this is a really good leaving group. So this OH is going to attack this carbon, and when that happens, you get the electron flow, you get that intermediate like we got before. Then that's going to, so you get that negative charge intermediate, which is then going to collapse back. So the electrons will flow back down, reforming this bond, and you're going to get a leaving of this group. So now you've lost that oxygen. So going back to our very first thing about this reaction being a condensation or dehydration reaction where you're actually losing a water. So we're losing our water. See, we're losing that oxygen, this oxygen. This is the oxygen we're talking about. This guy is already like hooked up. This guy's already lost it, and it's attached to this growing chain. So it's happening because these steps are separated, you're losing this group like way before you're actually going to add one onto this, because remember, this is the incoming and we just lost the oxygen from the incoming because we're hooking it up to a new oxygen with the transfer RNA. So it's kind of more confusing trying to figure out where exactly the hydrogens are coming from, because as I've talked about before, like hydrogens, so like protons can come and go. So like H+, they can leave and come and leave pretty readily, and depending on the pH of the solution and the pocket of the enzyme and all of these different things. So I'm not going to try to track the H's very much, which is why I haven't drawn them in color. But you do lose the equivalent of 2 H's, so depending on like what you start with, but because they can be in different protonation forms, but so just don't worry about what the H is really, or you're going to drive yourself crazy. So so now you're left with, so now you see you've lost this oxygen. So this oxygen is now part of this AMP. So this is AMP. So we started with ATP. We added this, we added the, this AMP onto here, and now the AMP is free. And we have this linked to this tRNA. And so this is going to be our incoming tRNA. And so now when you have this, so we have this codon, anticodon, this is all going to be from now on, this is going to come and bind here. And now everything's going to be taking place in a ribosome. So this big protein RNA complex. And so there's actually, the ribosome has like several sites. So you have like these 3 sites you can think about. So you have the P, where's the growing peptide, growing chain. The A, this is where it's going to come in. And the E, this is where it's going to exit. So our incoming one is going to be in this A site. The exiting one is going to be in the E site. And the growing chain is going to be in this P, in which it's so P for like peptidyl transfer, because that's where the transfer is going to occur. And when I talk about transfer, so we'll have the old tRNA, and it's going to have this growing chain. And then we have our incoming tRNA, and it's only going to have this single amino acid. And what's going to happen is this whole chain is going to get transferred onto this new tRNA. So this is going to be our old tRNA, which has the growing chain. And this is going to be our new tRNA, which has just the 1 amino acid. Okay, so this is the incoming. I'm just using old and new. So we're going to need to do that transfer, and that's going to happen in the ribosome. And so this transfer, okay, so we have the old tRNA. It's going to be attached similarly to how we have the new one attached. So it's going to be attached to the tRNA site. And what we're going to have is we're going to have this reaction where it's going to be so when it's in the enzyme, it's actually going to be, when you have this reaction occurring in like a pocket on like in the active site of the ribosome. So instead of the bulk water solution, you have this pocket where you're actually excluding the water. So there's not as much water in there. And when you have less water, so NH3+, basically charged things in general, water is pretty good at stabilizing them, because water, it has the partial negative and partial positive parts. And so it can put its partial negative parts, it can orient itself so that it's kind of stabilizing the positive charge of the nitrogen. And so this is going to make this more stable. So that's less likely to deprotonate. However, if you remove the water, now this is more unstable and it's easier to deprotonate. and you also have this ester linkage is helping to like pull electron density and to lower the, so you're basically lowering the effective pKa, so it's going to deprotonate more easily when it's in the pocket of the active site versus when it's just free floating around. Just remember, we need to have this in the deprotonated form, so it has this lone pair of electrons that can act as a nucleophile, and they're going to attack this carbonyl carbon, so similarly like to what we had before. So they're going to attack, and this is going to generate this tetrahedral intermediate like we saw before. So this Świderek ionic tetrahedral intermediate. And so tetrahedral referring to that this carbon is attached to 4 things. And Świderek ionic meaning that you have, it's overall neutral, but you have a separation of 2 charges. So we have a negative charge on the oxygen and a positive charge on this nitrogen. And so this is an unstable intermediate, and it's going to collapse down. So these oxygens will come back to reform this double bond. This, the bonds from here will go to this oxygen. The electrons from that bond will go to the oxygen. And now your old tRNA, it's freed from the old tRNA, and it's attached to the new tRNA. And the old tRNA is going to have a negative charge because you move that those electrons onto here. And you can see that, but you still have this, charge on the nitrogen. So the nitrogen is attached to 4 things, and so it doesn't have like 5. Like, so when it has a lone pair, it would have 1, 2, 3, 4, 5 electrons attached or assigned to it, and it likes having 5 because it has 5 protons. But in this state, it has 4, and so that's 1 less than 5, and so it has a positive charge because it has because it has those 5 protons. So it has this positive charge, and this is not going to be found in our peptide. And it's important that it's not going to be found in our peptide bonds because in the peptide bond, you actually have resonance stabilization. So this lone pair, when you have a lone pair, when you have this lone pair, you're going to actually have resonance stabilization between these. So you have partial double bonded character of both of these with the sharing of these electrons, and that's going to provide stability, and it's going to provide for the backbone. It's going to make the backbone like sturdier and limit its rotation. So you're actually going to end up kind of with a plane of planes. And depending on how bulky the side group is for the amino acids, you can get various degrees of rotation around those planes. And this is going to allow proteins to form like characteristic secondary motifs like helixes and sheets. But to do that, we need to deprotonate this nitrogen. And this actually happens, it's believed to happen through this like proton shuttle mechanism. And so if we zoom in on the old tRNA, so now this, now I've kind of flipped this over, sorry, but so if you look at this, like, so this was our 3' OH, so this is now this, and so our 2' OH is here. Yeah, so I was basing it, off of a figure, but that figure was drawn differently than other figures, and so hopefully I got everything okay. But this is your 3' OH, and you would think that this would just like pull off that proton, because negative positive, this one would take that H, but it turns out it's more complicated because in like structures of this, you can see that this is too far away to pull that off, whereas the 2' OH is near enough to pull it off, but the 2' OH is already protonated. And so it turns out they think that the OH, so that this oxygen is actually going to take from this coordinated water. So there's a water molecule here too. So if you're looking at that paper, you might get kind of confused because there's a water molecule sticking out like this, and you're like, what the heck is that? Because it's all drawn the same color there. That's a water. And so this can pull a proton off of that water, and then that gives you OH minus. OH minus can then take a proton from here. So this one becomes O minus. Then that can pull a proton from here. So you have this very complicated way to get this back to its protonation step. But so it's called a proton shuttle. And so now you're left with, you have your tRNA, you have that regenerated. You have your peptide bond with the ability to have that resonance stabilization. And now everything is attached to this new tRNA. And then with the help of like elongation factors and things, this is going to this all shifts into the P site, so where the growing chain is on your amino acid of your ribosome. So this is going to shift and be in this, the P site, the growing chain, and then a new one's going to come back in. So overall, we have the same end product as we would see if we just looked at the basic overview of the reaction, except that we are doing this in a sort of convoluted way, taking these different steps in order to first, so activate the amino acid, so give it the energy it needs, make that make that carbon more reactive, and by adding this better leaving group. Then we're going to charge the tRNA, so this is going to give us the specificity that we need, going from, so that the right amino acid is added that corresponds to the mRNA. And then finally, we're going to, with the help of the ribosome, and we're going to facilitate this reaction and make it happen. Another cool thing, so this is showing you like the tRNA that's helping, the old tRNA, how it's helping with this proton shuttle, is, so it turns out that if you have, because this OH, this 2' OH is participating in this shuttle, this reaction is not going to work if you have the DNA there, so if you have a deoxyribose there. So I thought that was pretty cool. So I'll link to a couple papers I found when I was trying to look into all this further, and sorry if I misdrew anything. So I just trying my best, and some of the stuff I'm a little rusty on, but trying to get better. And so I hope that this was helpful to someone. And if I did make any mistakes, please let me know. So I really don't want to be teaching anybody anything wrong. But it's hard when you look things up because there's always complicating factors and people arguing about which way it really is. But hope this helped.